right, now we're going to look at chapter 22, the nervous system diseases. Now, one thing we want to look at before we start talking about the diseases is we want to look at some terminology, but we also want to look at how the nervous system is divided into different areas. So if you'll notice here, we have the central nervous system. The central nervous system is centrally located, and this is going to include your brain and your spinal cord in the center part of your body. The peripheral nervous system is the stuff out to the side, and this is going to include all of your peripheral nerves who are going to help with motor movements as well as sensations. So when we look at some of these terms, there is something that's called meningitis. Anytime we add itis to the end of a word, it means inflammation. So meningitis is inflammation of the meninges. The meninges are special coverings that are around your brain and your spinal cord. They're there for protection to help with that delicate tissue. We then see encephalitis. Encephalitis is inflammation of the brain tissue itself. Now, therapy here, guys, when we look at chemotherapeutic drugs, they need to be lipid soluble if we're going to use them for the nervous system. The nervous system has a special barrier around it that is called the blood-brain barrier, or the BBB. This barrier is going to filter out what gets to go in and out of the structure to the brain. It's kind of, think of it back in the old days, back when there were kings and things like that, and they would have cupbearers. The cupbearer would have to eat the food or drink the drink first, and if it was poisoned, they would die, and then the king would be saved. We see that that's kind of what this blood-brain barrier is there for. It's helped protect the brain from potential toxins. But because of this, it also limits certain medication from getting into the brain as well. And so anytime we're looking at chemotherapeutic drugs that are going to deal with the brain or the spinal cord, they're going to have to be lipid or fat soluble in order to cross that membrane. And one example of this is the chloramphenicol. All right, so looking at meningitis, meningitis has some general symptoms that are going to be present. These include fever, headache, and a stiff neck. It can progress, though, to more severe things that can be convulsions and then a coma. When we get to this point of the convulsions and the coma, they could suffer from neurological damage or even death. Now, there are different types of meningitis. There is viral meningitis, which tends to be more common, but it is also going to be milder in its effects. Bacterial meningitis is a little more severe. Death of the bacteria causes the release of endotoxins. These endotoxins then can result in the patient going into shock and having extreme inflammation. These particular types of bacteria are normally gram-negative microbes. Now, there are some vaccines available for some of the strands of meningitis. This is to help reduce the incidence of the cases of meningitis. Some of these examples are Streptococcus pneumoniae, Haemophilus influenza type B or HIB, and Neisseria meningitidis. So in order to diagnose what kind of meningitis is present, we would have to do a cerebral spinal fluid diagnostic test. So the CSF diagnostic testing. We would do serological tests followed by a culture. So when we look at the serological test, this can be done under an hour. However, the cultures can take 24 to 72 hours. So in the first 24 hours, it would give us the preliminary type of results. However, in the 72 hours, we would have the final results of what potentially is causing that infection. Now, this is important to process the specimen quickly because a lot of these pathogens cannot survive well once outside of the body. So it's important that we collect it and we transport it in the proper way. Now, the way that we get the cerebral spinal fluid is through what we call a spinal tab. So the individual has to lay on their side in the fetal position. This opens up the vertebrae a little bit in order to insert the needle and collect some of this cerebral spinal fluid. In the first tube, they collect it for chemical analysis. So they're going to look at the glucose levels, protein levels, the chemicals that are present. The second tube, they're going to look at the hematology. So this is going to be looking at if there's blood present, white blood cells, things like that. And then the third tube is going to be the tube for the microbiology for the idea of the cultures. Now, there are some rapid tests, but these rapid tests don't give us the exact results. A negative test does not exclude the possibility of bacterial meningitis. So we can say, oh, it might not be, but we need to wait for the culture test to come back in order to definitively say, no, this is not bacterial meningitis. So with bacterial infections, we see that there is the bacterial meningitis. They are going to have capsules around them as well as producing toxins, and we just hit on these three, and we'll talk a little bit more about them in a minute. We also have, in the bacterial category, Listeria monocytogenes, Clostridium tetani, Clostridium botulinum, and Mycobacterium leprae. So let's look at the Streptococcus pneumoniae first. This is a nasopharyngeal normal flora, so most people are carriers. Actually, about 70% of the population are. And it is a normal type of flora in the nasal or throat area. However, if it gains access into other areas of the body, it could cause pneumonia if it goes deeper into the lungs, as well as a middle ear infection if it gets into the otis media. We see that this is today's leading cause of meningitis. The mortality rate is higher in children. That's about 30%, but then in the elderly, it's about 80%. One thing that's really kind of helpful with this types of vaccine as well is it does help prevent meningitis, but it also helps reduce the number of ear infections for individuals as well, since this is the main culprit for ear infections. All right, so if you look here, you can see the Streptococcus pneumoniae growing on the blood agar plate. We also see that this is showing you what the meninges are. This covering that is covering the brain that they're kind of pulling away with the tweezers is the part that gets inflamed, and it can put pressure on the brain tissue itself. So when we're looking at Streptococcus pneumoniae, it is a gram-positive bacteria. Therefore, we see that it does stain in a purple color. It has the lancet shaped. It's called a diplococci, so it's two cocci kind of put together. It is alpha hemolytic, meaning it does use some of the red blood cells. It's able to do a little bit of hemolysis. Also, we'll notice that it is encapsulated. It has this capsule around it, which helps it evade your immune system. Since there are different types of alpha strep, we want to see if we're dealing with Streptococcus pneumoniae or not. And so we can do what we call the optogen testing. Again, this is a type of disc test. We see here in this case, if the disc does cause an area of no growth, the area of inhibition, we do see it's Streptum pneumoniae. Other alpha strep do not have that zone of inhibition. Haemophilus influenzae is a normal throat flora. Again, a lot of times we are carriers of this. It can cause, though, pneumonia if it gets deep into lungs, as well as ear infections. We see that this H. influenzae type B is known as the Hib meningitis. This is historically the leading cause of meningitis in children. Today, though, it is on the decline due to effective vaccines that are available. So in the lab, if we're going to take a look at this particular type and we're trying to see what kind of meningitis the patient has, we would see that these are going to be tiny gram negative bacteria. Okay, so unlike what we saw with the strep, those were gram positive. These are going to be gram negative, the pinkish color. These are also what we call coccobacillus. All right, so they are kind of in between the coccus shape as well as the bacilli shape. These are very fastidious type of bacteria, meaning that they are going to be very picky. They are also blood-loving bacteria. They require two distinct growth factors in order for them to grow properly. Okay, we see that it can be represented here with this V or the 5 growth factor, and we see that's in AD, and then the X or 10 growth factor, which is the hemin. We need both of them in order to grow. So if you look at this plate, they gave no factors at the bottom, so there's no growth. They gave growth factor X on one side, growth factor V on the other, and we still see no growth, but when we give them both, growth does take place. Okay, so these are very picky bacteria. They need certain things present in order for growth to happen. When we're also looking at this group, Haemophilus influenzae, they do prefer the chocolate agar plates, the laked blood, and the reason they like this is it does provide their two factors that they prefer. Okay, they're not going to do the hemolysis in the sense of breaking the red blood cells apart, but if they're already broken apart in the chocolate agar, they gain access to what they need. However, these plates do smell like dirty socks. Okay, when they're growing on it, it does smell like dirty socks. There can also be Haemophilus saddling around beta hemolytic staph aureus on blood agar, because they are able to actually do hemolysis of the red blood cells. So when they do hemolysis and they release all the contents of the red blood cells, it gives those two factors that this particular bacteria would want. So it causes satelliting or growth around the beta hemolytic bacteria. The next group is the Neisseria meningitis. This is going to also be found in your normal flora of the nose and the throat, and about 40% of the population are carriers. The symptoms that happen at this meningitis are due to an endotoxin production. So we see that there can be a quick onset with a sore throat, which then can lead to septicemia, which then leads to meningitis pretty quickly. The people or the groups who are at risk are children under the age of 2 years of age, as well as college students and historically the military. military. And the reason why college students in the military have been a potential risk population is due to the close contact when individuals live inside of dormitories. All right, so they're in close contact with people, and so it can potentially spread very quickly. Now, there are vaccines available for some of the serotypes of this particular group, but not the childhood types. Okay, these are going to target particular components of the cell walls. That's what the vaccine is going to do. So it does target type A, C, W, X, and Y. However, it does not target the serotype B, which is the childhood type. So if we're growing this in the lab, we see that it is also a gram-negative bacteria. This is going to also have a diplococci type of shape, or more like a kidney bean shape. We see that when it grows on the agar, the blood agar, you can see it here, and having kind of a whitish type of colony. We see that it is aerobic, meaning it needs access to oxygen, so it does come up as oxidase positive. And then we can also do what we call the cysteine tryptic agar test, or the CTA. This is a type of sugar test. Okay, what we do is we take these different broths, and they have different types of sugars present. Okay, we see that there's glucose in the first one, the second one has lactose, the third has sucrose, and the last one has maltose. Now, if the bacteria can utilize that sugar, it's going to cause the color to change from red to yellow, and you can see that here at the top. So these guys can use glucose and maltose. They are positive for that, but they cannot use lactose and sucrose. All right, and so you can see the two ends have the yellow on top, which shows you they're positive. The two in the middle still have the red color. Listeria monocytogenes is going to be a gram-positive bacillus. This one can thrive in cooler temperatures, therefore it can survive inside of the refrigerator. This one causes neurological disorders in stillborns. Okay, the at-risk types of populations are the immunocompromised adults. This can be a food-borne type of pathogen, which then causes sepsis and meningitis. With fetuses and newborns, it can actually cross the placenta. Okay, so when we see here that the mother may not be affected too much by it, but it can cross the placenta and cause an abortion to take place due to the infection, or it can also cause meningitis, which could lead to mental retardation in or the newborn. Okay, and so this is one of those that there are a lot of precautionary things that are told to pregnant women about not eating certain things because listeria could potentially be present. One example is lunch meats, okay? Because where do we store lunch meat? In the refrigerator. They could be there. One way to take care of this is to heat the lunch meat up. So it's not saying you can't eat lunch meat at all. You should just heat it up. Another common cause of this is found on melons, like cantaloupes or watermelon. Okay, normally when we eat fruit, we wash the outside. However, a lot of us don't wash the outside of a cantaloupe or watermelon because that's not the part we eat. However, if you don't clean the outside and you use your knife and cut through it, it can then take the listeria from the outside into the watermelon or cantaloupe, which then means it could be passed on. So when we see here with listeria, these are going to be found in some food products. Their reservoir is found in animals, and we see that there is the fecal oral route that takes place. Our food gets contaminated from feces. This can happen due to poorly pasteurized dairy products. So some of the poorly pasteurized dairy products could contain the listeria. Foods in contact with raw beef, pork, or poultry goods. Okay, and so making sure that everything has been cooked really well. And then, of course, some of our raw vegetables can also carry the listeria. Again, for an adult, a normal healthy adult, the symptoms for listeria are very mild. You may feel like you kind of have a cold. However, for the fetus or newborn babies, it does have a high mortality rate. The next one we have is Clostridium tetani. This is also known as tetanus or lockjaw. This is where deep wounds with spore-contaminated soil is going to be the issue. So a lot of times we talk about it being where you've been cut or stepped on like a rusty nail. Okay, when we look at that, the nail penetrates through the skin, allowing then whatever was on the surface of it to be able to gain access inside. And if this particular bacteria is found on that metal structure, then it can cause this illness. Symptoms are from a potent neurotoxin. They create an exotoxin that they release. This enters into the central nervous system via the peripheral nerves. So the peripheral nerves are going to carry the toxin from the outside part towards the spinal cord and the brain. This creates very severe muscle spasms, okay? And so it causes the muscles to contract, kind of like having a Charlie horse, but one that does not relax, okay? It's hard enough when you get that Charlie horse in your calf or something like that, and it doesn't relax for a long time. This would mean that it won't relax due to the toxin. Now, this can cause the individual to need supportive respiratory care. There's a big muscle that helps you breathe. It's called your diaphragm. If the diaphragm contracts and cannot relax, you can breathe in, but you cannot breathe out. So we would need to help the individual be able to breathe. We also see there is a toxoid vaccine that is available. This is found in the DTaP or DTaP vaccine. This contains a vaccine for diphtheria, tetanus, and pertussis. Okay, and so a lot of times if you need a tetanus shot, this is one thing that they're looking at. This stimulates antibody production that neutralizes the toxins. It actually doesn't attack the bacteria or kill the bacteria, but it neutralizes the toxins that they do produce. You do need a booster of this about every 10 years. Now, some other treatments that we could use for tetanus are antitoxins called TIG. These are borrowed antibodies, but we also then need to do wound debridement. We need to get that wound cleaned out, get the contaminated soil and the bacteria out of there in order to reduce the amount of toxin that's being produced. So if we take a look at this, Clostridium tetani is a gram-positive anaerobic bacillus. Okay, so gram-positive, it's the purple color. Anaerobic means it does not want oxygen. This is why it needs to get deep into your tissues. It does have kind of like a boxcar type of shape. They can also contain terminal endospores. Now remember, endospores are a way for them to be able to survive for long periods of time in adverse types of conditions. All right, so they can be found on that nail, and that they could be on that nail for years and years before they gain access into a person. Now this is going to cause major muscle spasms due to the neurotoxins. It causes a contraction without relaxation, and some of these contractions can be so severe that they can even break the bones of that the muscles are attached to. All right, the next group we have are the Clostridium botulinum. This is also known as botulism or food poisoning. A lot of times the culprit of this is poor canning techniques. If we do not can things properly, it actually promotes the growth of this anaerobic type of bacteria. Clostridium botulinum also produces a neurotoxin, an exotoxin, but this one interferes with the neurotransmitter acetylcholine. So instead of causing the muscles to contract, it causes them to stay relaxed. It makes it where the nerve cannot talk to the muscle to allow it to contract. Now, they can also produce endospores. These endospores are heat stable, meaning that they can survive that heating process. Ingestion causes really no harm of these guys. Exotoxins, though, can also be the exotoxins are heat liable, meaning that they can break down if you heat that up and you denature the toxin. Now, botulism antitoxins are available. So this particular Clostridia botulinum is a gram-positive anaerobic bacillus. It has terminal endospores that can be produced. It is commonly found a lot of times in natural honey. This can cause infant botulism. This is why you do not feed honey to children who are under 1 year of age. The endospores will multiply in the intestines and start to produce the neurotoxins, and the child's immune system cannot overcome that. We also see that we can have wound botulism, where this gains access, causing issues in a wound. One thing we do use this bacteria for is with Botox. We take the toxin that Clostridium botulinum uses, we then use it for cosmetic reasons. Okay, what it does is it causes those muscles in a sense to be paralyzed. It causes them to not be able to contract, reducing the formation of wrinkles, but these people also don't have any facial expressions normally because of that. And it also reduces perspiration. All right, and so this is one thing that we use. Now, what we're only injecting is the toxin, and we're doing it in very low amounts, and so it will eventually wear off, and this is why Botox treatments have to continue. We are not actually injecting the bacteria into the patient. The next group is the Mycobacterium leprae. This is also known as leprosy. You probably have heard of leprosy if you've ever read like the Bible, or when it talks about in like ancient times where it hit on leprosy. This is a big thing. We also see it's known as Hansen's disease, which is a little less commonly used and also not as scary to kind of talk about. Leprosy does grow in, when we talk about Mycobacterium leprae, it grows in the peripheral nervous system, and this results in nerve damage. It does prefer cooler temperatures, around 30 degrees Celsius. And so this is why it prefers the peripheral nervous system versus the central nervous system. Central nervous system is more centrally located, which means that it's going to have the higher body temperature. People who are contaminated will actually secrete the bacteria or the organisms from their nasal mucosa, and it can then infect other people. It's not very contagious. You actually need really close contact for a prolonged period of time to get this. However, they thought it was a lot more contagious back in those old times, and that's why those people were in leper colonies where they were separated, okay, because if they lived with you, there was a higher chance that you would have that contact and get it. And also, this is why they would say that they were unclean as they came through areas where other people were located. They may even wear bells and things like that to let people know that they were coming into the area. Now, this is very difficult for us to grow in the lab. A lot of times it has to do with the cooler temperatures that are present, and so we actually have to use more of the tissue cultures. One thing that we do use is going to be with armadillos. Armadillos actually have a lower body temperature, and so they're a good source to be able to grow this bacteria on, as well as the foot pads of nude mice. Now remember, we're going to use nude mice because if they have the fur on them, this is going to cause it to be too warm, and the bacteria would not grow. This bacteria was isolated for the first time in 1870, and when we're talking about its growth in the lab, is it actually takes 12 days for it to replicate. So this is a very slow process of going through, of seeing this bacteria being grown in the lab. We also see that it has the acid-fast positive type of staining. All right, so we would use that acid-fast type of staining, which is very similar to the Gram staining, but we would use the different chemicals that we discussed way back in chapter 4. So when we're looking at the Mycobacterium leprae, there is what we call the tuberculoid form. This is where you have 4 or less lesions. However, it can then progress into the lepromatosis form, and this is going to be where you have decreased cell-mediated immunity. It causes septicemic multi-organ failure eventually. But if you'll notice, guys, the things that are affected first are areas on the peripheral structures. So they'll start to lose their fingers, toes. It causes a deformity of the face and things like that, where it starts to then progress inward. Again, though, in some countries like India and even others, there are still leper colonies where people who are diagnosed with leprosy are sent to live. We do see that there is a vaccine that can help with protection against Mycobacterium leprae. It's the BCG vaccine. This is a vaccine that we normally use for tuberculosis, but it can also help give some protection against leprosy. All right, so now we're going to look at the viral diseases of the nervous system. With the viral diseases, these include things like polio, rabies, and arboviruses. These arboviruses are arthropod-borne encephalitis. A lot of times this is going to be a group that are the East and West equine encephalitis, West Nile, and some other viruses. So let's look at polio first. So poliomyelitis is caused by the polio virus. It is an RNA virus, meaning that it has the RNA strand, not the DNA. So this is going to have where it has the bases A, C, G, and U. This is spread by ingestion of contaminated food or water, okay, and a lot of times it causes just intestinal distress at first, but then it starts to progress to the central nervous system, which then can result in paralysis. Now, prevention, when we're looking at the poliomyelitis, we see that it can be an inactivated virus vaccine that we call the Salk vaccine. This is where we're going to inactivate the virus and then inject it into the individual so that the immune system then can fight a dead form of the virus. There's also what we call an attenuated virus oral vaccine known as the Sabin vaccine, and this is where it's going to be a weakened form of the virus, and instead of giving it in a shot form, it's going to be in a liquid form that then the child needs to drink. So when we look at the intestinal distress, a lot of times it starts as a sore throat. It progresses then with fever, nausea. It then can progress to the central nervous system in about 1% of the cases. Now, when we're looking at the inactivated virus vaccine, it is a series of injections that the individual has to have. And so, guys, when we're looking at this prevention, a lot of times it is a series of injections or vaccinations that do need to occur to offer protection. Now, a lot of times there are individuals when polio was a lot bigger of an issue in the United States or in even other countries right now, there is something that's called post-polio syndrome. This happens with individuals who have actually survived having the polio virus. However, their muscles are weakened, and sometimes we don't even see it until they are a little bit older. It's a weakened nervous system connection with the muscles, and this is a slow progressive process. A lot of people talk about how, well, polio is not around anymore, so we don't need to be vaccinating. The reason why we don't see polio very much in like our country of the United States is because of vaccinations. In other countries where vaccinations are not readily given or available, we see that polio is still a major issue. All right, so the next one we want to look at in the viruses is rabies. Rabies is going to proliferate in the peripheral nervous system. It's going to start there, and it will eventually spread to the central nervous system. We see that it is an issue because we have a poor immunological response. Our immune system does not respond very well to rabies, and one reason is because it gains access into the nerves. Recall we talked about this back when we talked about chickenpox and shingles. Once the virus gains access into the nerves, it's very hard for your immune system to find it and attack it. Now, rabies is actually the Latin word for rage or madness. Okay, that's what it's kind of talking about here, rage or madness. And it does cause the individual to start to go, in a sense, mad or crazy. It causes fatal encephalitis. The brain starts to undergo inflammation, which then causes death. Now, the virus can be transmitted by the infected saliva through a bite or a wound, and this bite sometimes can come from different organisms. A lot of times we talk about it being dogs. Dogs can have rabies, but so can a lot of other animals like fox, skunks, squirrels, things like that. One issue we see with rabies is that sometimes individuals will get this without having a bite or a connection with somebody or something that has been infected with the virus until they realize it was in transplanted tissues. Rabies is one of those that's really hard to detect that the individual has died from that, and so because of that, sometimes some of their organs can then be given off for transplants. One particular one is with the corneas in the eye, and it's transplanted to a new person, which then the rabies virus then infects them. Okay, we see that this has happened before, and so it needs to be tested, okay, to make sure that the rabies virus is not present. The next one is the arboviruses. The arboviruses are normally going to have a reservoir between birds and the mosquitoes. That's where they're going to normally go back and forth to. However, when the mosquitoes bite somebody else, when they have been infected with it, they can either bite horses or humans. It can be transmitted to them as well. It is what we call a mosquito-borne virus. It does cause encephalitis, okay? It does cause inflammation of the brain. Now, there are different types. There are Eastern and Western equine encephalitis. Okay, so Eastern and Western equine encephalitis. We also see West Nile virus. West Nile virus was first seen in the United States in 1999. It causes chills, headaches, fever, and then it can progress to mental confusion and then to a coma. We see that the cases of these viruses increase in the summer months, and this is due to the fact that people are outside more and the fact that the mosquitoes are going to be more active at this time as well. All right, so now we want to look at the fungal diseases of the nervous system. So the first one here is the Cryptococcus neoformans. These are going to be able to be identified using a negative stain with like India ink. They form large capsules around them. We can use differential media to grow Cryptococcus. The dark colonies would be the Cryptococcus neoformans. The light colonies would be the Candida albicans, which is a different type of fungus. Cryptococcus is going to be linked to pigeon issues. We are exposed to this through their droppings. When pigeon droppings are found and you go and sweep them up or things like that and you breathe in the particles, they contain the fungus and can ultimately make you sick. Now, the reason why this is such an issue is that pigeons produce about 25 pounds of droppings per year, and so the more pigeons you have in the area, the more droppings are present, which increases the risk of potentially coming in contact with this fungus. We do see more people are more susceptible where they don't just have the cold-like symptoms where it can progress into a more life-threatening type of infection. These would be people who are immunocompromised, like with HIV patients or AIDS. We also see that it can progress into meningitis, and ultimately this increases the mortality rate if that does occur. So now we're going to talk about the protozoa diseases of the nervous system. So the first group are the trypanosoma. These are a type of protozoa, and they cause something that's called sleeping sickness. This is going to be carried by the tsetse fly. We see that this is what the trypanosoma looks like, showing you in red blood cells, and these are going to be flagellated type of protozoa. The problem with this protozoa is that it hides in plain sight. Your immune system recognizes that it does not belong, so it starts to fight the invader. By the time your immune system gets a good defense against it, it has changed what it looks like. And it looks like a different invader. The protein coat looks different. So then your immune system is like, hey, that doesn't belong, and it starts to try to fight it again. And by the time we get a good defense, it's changed what it looks like again. And this can continue to occur. This is one reason why it's called sleeping sickness, because the individual, when you're sick, if you think about it, when your immune system's in overdrive, you feel tired. The immune system is continually doing this, which causes the patient to feel more and more tired until they finally go into a coma, and it does result in death. And this occurs once the central nervous system is affected. Now, there is a drug that can be used to help treat African sleeping sickness. The problem with this is the drug is very highly toxic, and it kills 5% of the patients itself. All right, and so it's very, very toxic, and so you're taking the chance by giving them the treatment as well of it potentially killing them. So Naegleria is another type of protozoa, and this one's an amoeba, so it's going to move by having extensions where it can pull itself forward. These are going to invade through your nasal mucosa membrane, so through your nose, and then it will ultimately move up to your brain and start to feed on your nervous tissue. There is a 100% mortality rate due to a lot of times the misdiagnosis. This is not very common, so most of the time the patient is misdiagnosed, which leads to their death. And we normally find the protozoa during the autopsy. This can enter in, and obviously it causes encephalitis of the brain. This is going to be found in contaminated water. Okay, and this water is going to be things like rivers and streams, stuff like that, that people potentially may be swimming in. Okay, and so this is just one example. All right, this group is the Prion's diseases. This is what we call the Creutzfeldt-Jakob and Kuru. This is going to be caused by a misfolded protein. All right, and so it's not even really a viral infection. It's a misfolded protein. This is going to cause the other proteins in the individual's brain to start to misfold to where they're not doing their job, which causes the brain to start to shrink and deteriorate. And this can happen very rapidly, or it could happen over a series of years. So the Creutzfeldt-Jakob disease is going to have a classical and a variant form. This could be genetic, where it's passed down from individual to individual. The Creux is due to cannibals. There are some cannibals that are still present. These are individuals who are not going to eat you if you happen to go onto their island or into their habitat. This is normally due to religious reasons. They believed that in order for their ancestors to pass on to the next life, whatever it might be, they need to eat the brain and the heart of that relative. The problem is this prion is in the brain, and then it gets passed on from one person to the next, and it can continue that process. Gives yourself a death sentence whenever you consume that tissue. Because guys, these are not heat liable, meaning these proteins are not hurt by heat. Most proteins will unfold and stop doing what they're doing once they've been heated to a certain amount. These guys are not. And so even if you cook something really well and that sort of thing, it still could potentially cause the disease, the problem. There's a last group here in this particular chapter that's called the miscellaneous diseases. This is called chronic fatigue syndrome. This has pretty much an unknown cause. This is why it's not really labeled under viral, bacteria, fungal, that sort of thing. However, it causes persistent fatigue, and it is also linked to allergies. This is going to be where 6 months at least before you are diagnosed with this. You have to have 6 months of this fatigue before you're diagnosed. There's a list of symptoms that are found on page 633 in your textbook. All right, so this covers the nervous system. Again, if you have any questions or concerns, please let us know.